So this is going to be the exam 2 review. We're going to be going over the practice problems for the exam 2 practice exam, of course. So we're just going to go ahead and jump right into it. So question 1 is asking a solution with a pH of 4 has a blank concentration of ions than a solution with a pH of 8. So I'm not a huge fan of this question because it's a little misleading. When we're talking about ions, we're not talking about sodium. We're not talking about chlorine. We're talking about a very specific type of ion, and that is a proton. So what is a proton? Right? So a proton, we just have hydrogen. Hydrogen has, of course, a mass of 1 and an atomic number of 1. So what does that tell us? That tells us that hydrogen is just a proton with no neutrons with 1 electron. Right? So that's a proton. Sorry. Well, what I drew here, that's going to be. Sorry, I just need to get my bearings a little bit. So, when we're talking about hydrogen, right? This is on the periodic table. Hydrogen has an atomic number 1, weight of 1, so that tells us that there's no neutrons, just a proton with a electron orbiting it. So this would just be a regular hydrogen atom. But when that electron is lost, that electron goes away, right? We don't see that anymore. When it goes away, we're losing negative charge. So this is going to have a charge of +1. But there's nothing else. There's no electrons, there's no neutrons. This is just a hydrogen proton. So we just shorten it by calling it a proton. If that was confusing. Right. So now that we got the language sort of out of the way, we could start talking about pH. Right? So when we're talking about pH, we're not talking about any other ion other than protons. So here we're talking about protons, right? Which we just discussed. So solution with a pH of 4 versus a solution with a pH of 8. So what does that mean? Right? So the pH scale, right, it runs on a scale from 1 to 14. Sorry, it runs on a scale from 1 to 14. So 7 being neutral, 14 being basic, and 1 being acidic. So, when we run this way, that's becoming more acidic. And when we're running this way, that's when we're becoming more basic. So what does this mean? Right? When we're talking about pH, this P, we don't really recognize the P. P doesn't really mean anything. What we're really focusing on is the H portion of pH. So whenever you have something in brackets like this, that just means concentration of. So when we're talking about pH, we're talking about the concentration of protons in a solution. So when we have concentration of protons in a solution, that's the measure of how acidic or basic something is. So when we have a very high concentration of protons, that's when something is acidic. So acidic, high concentration of protons. And now when we're talking about basic, that correlates to low concentration of protons. And the further you go this way, the more basic something is, the lower that concentration is. And vice versa, the further you go this way, the higher the concentration is of protons. So that's basically the gist of the pH scale, right? Another thing that you don't really have to know for this exam is that the pH scale runs on a scale of magnitude. So if we have something with a pH of 8, Something with a pH of 8 is going to be 10 times more basic than something with a pH of 7. And something with a pH of 9 is going to be 10 times more basic than something with a pH of 8. And 100 times more basic than something with a pH of 7. Or what if we have something with a pH of 2? Something with a pH of 2 is 10 times less acidic than something with a pH of 1. And something with a pH of 3 is 100 times less acidic than something with a pH of 1. So, like I said, on a scale of magnitude. So, not that you really necessarily need to know that, but just good information. So, anyway, going back to the problem is asking a solution with a pH of 4 has a concentration of protons, then has a blank concentration of protons, than a solution with a pH of 8. So what is this telling us, right? So pH of 4, that's 4 is less than 7, of course, so that's going to be acidic, right? Versus something with a pH of 8, that's going to be basic. We just learned this, of course. So acidic versus basic. So what's going on here? The question is asking whether it has a greater or lesser concentration of protons. So something with a pH of 4, as we just discussed, something that's acidic has a higher concentration of protons, right, than something that's basic. So something that's basic has a much lower concentration of protons. Whereas when something has a pH of 7, it's sort of like equal. It's equal with a different molecule known as OH, or rather hydroxide. But we don't really want to worry about that. We're just focusing on proton concentration. So, of course, taking all of that into account, the answer would be A, greater. So a solution with a pH of 4 has a greater concentration of protons than a solution with a pH of 8. And we know this because the more acidic something is, the more protons it has. The more basic something is, the lesser amount of protons it has. So that's question 1. We can go ahead and move on now to question 3. And we're moving on to question 3 because on the practice exam, question 2 has just been sort of marked out because we don't really care about tetrahedrons and the shapes of things in this class. So yeah, just moving on to question 3. So question 3 is asking, which of the following is not a common element found in organic molecules? So, as I'm sure you may know by now, there are 4 major organic molecules in humans, and that, of course, are carbon, oxygen, hydrogen, and nitrogen. So there are everything that's not this makes up like 98% of organic molecules, and especially the chemical composition of molecules in the human body, or even beyond the human body, beyond any living thing. So this is a large portion of the composition of organic molecules. So anything that's not this is not commonly found, right? So anything that's I don't really need to go further than that. So let's look at our answer options. Carbon, hydrogen, oxygen, sodium, nitrogen. So carbon, hydrogen, oxygen, nitrogen, these are all molecules that are found in our bodies, right? To a great degree, rather. So what's the answer? Sodium. Sodium is not a common element found in organic molecules. That isn't to say that sodium serves an important role and that sodium isn't used, but sodium is not common. It doesn't consist of 98%. Anyway. So, moving on to question 4. So, I've been getting a lot of questions about functional groups, right? So this has to do with functional groups. So question 4 is asking, hydroxyl groups are the primary chemical group found in a particular biological macromolecule. Which of the following statements regarding this macromolecule is true? Okay, A, it is likely to form hydrogen bonds with water. B, it is unlikely to dissolve in water. C, it will likely dissolve in non-polar solvents, and D, is likely to have a planar structure. So first things first, we're just going to eliminate D. We're not concerned about the shape of molecules necessarily. So there are a few shapes that comes in molecules that's like bent, tetrahedral, planar. We're not talking about that. So just ignore D. So that leaves us with likely to form hydrogen bonds, unlikely to dissolve in water, and likely to dissolve in nonpolar solvents. So one thing about functional groups is that they're exactly that, right? They serve a function. They are groups within a molecule that serve a function. So what are these functions, for example? So let's draw out the functional groups that you need to know for this class. So we have hydroxyl, we have carbonyl, we have carboxyl, we have amino, we have phosphate, and we have two more: methyl and sulfhydryl. So just looking at these, right, the most important ones are hydroxyl, carboxyl, amino. So these are the big three, and then sort of the lesser three. Sorry, then we have the lesser two: carbonyl, phosphate, and then we have the ones that are pretty uncommon. So it's going to be purple: methyl and sulfhydryl. So sulfhydryl appears in one place in one macromolecule. So there are 20 amino acids, right? So one out of the 20 amino acids contains a sulfhydryl group. That's it. So we don't really ever want to worry about sulfhydryl. Don't even like concern yourself with sulfhydryl. How about methyl? Methyl is important, yes, but methyl doesn't really apply to any molecules or macromolecules that we observe in this class. But one thing that's important to note about a methyl group is that it's comprised of C and H. And what is C and H? That's a hydrocarbon. And what is a hydrocarbon? That's nonpolar, right? So if we see a lot of methyl groups act as a nonpolar group within a molecule, right? So let's look at the rest of these. Let's look at the rest of these, starting from carbonyl. So what does a carbonyl do? A carbonyl has, of course, a ketone, not a ketone. Sorry. A carbonyl, of course, has a C double bond to an O. So what does that tell us? This O has a partial negative charge on two ends, right? So because this has a partial negative charge, it is considered to be, of course, polar. So what can this do? It can go ahead and bond to water via hydrogen bonding, right? That's able to do. So whenever you see a carbonyl within a molecule, that's how you know, well, okay, well, there's a pole that's being created, and this pole can attract different molecules such as water or other polar substances like ethanol or propanol or other polar substances that you may or may not know. So that's the purpose of something like a carbonyl, right? It acts as a polar group. So how about a phosphate? So phosphates are often just abbreviated as a P with a circle around it, or with a P, or PI, standing for inorganic phosphate. So phosphates, the cool thing about phosphates is that they have these two negative charges, right? These two negative charges, what do these do? These negative charges can attract positively charged ions, aka cations, or they could attract something like water or other polar substances, right? They could attract the positive poles of water, which, aka, that's the hydrogens of the water. So that's what these can do, right? That's why something with phosphates something with phosphates are very polar. So what includes phosphates? Of course, that will be the nucleotides, and that'll also include phospholipids. So both very important, right? Both very very important for this class. So phosphates are, you don't see them as often, but they do exist in two very important things. So what's next? Hydroxyl, right? So when we're talking about hydroxyl, what is it? It's an OH, right? Hydroxyl can also be known as an alcohol, right? So hydroxyl and alcohol can be used interchangeably. They mean the same thing. But for the purposes of this class, we call it a hydroxyl. So, what's a hydroxyl? It's exactly that. Hydrogen and oxygen. What's a hydrogen and oxygen? That's a polar covalent bond. So what is this able to do? This is able to create a hydrogen bond with water, which is very important for creating a polar molecule, right? So this is able to form hydrogen bonds, and this is able to mix with water. So that's like the purpose of hydroxyl. So what has a lot of hydroxyl what has a lot of hydroxyl functional groups in it? Sugar. So you could dissolve sugar in water, right? I'm sure many of you do that when you make your morning coffee. So what does sugar look like? Sugar looks like this. So let me draw out sugar real quick. So how many hydroxyl groups do you count here? 5. So because it has so many of these polar groups, right? So what I drew here, this is glucose. Because it has so many polar groups, this is able to make a ton of hydrogen bonds. So it's considered to be very polar. So that's why you dissolve sugar in water and not in oil, right? So that's the function of a hydroxyl group, to make hydrogen bonds, because the only thing that it consists of is a polar covalent bond. So how about carboxyl and amino? So carboxyl and amino we could tackle at the same time, because both of these functional groups exist in one macromolecule that we care a lot about, and that is proteins. So when we talk about proteins, right, what does a protein look like? A protein has a very specific backbone that you need to know for the exam. So a protein has a very specific backbone, and then I'm going to draw in yellow the R group, because the R group changes, right? So a protein has a very specific structure to it. So a protein will always have an amino group. Did I say a protein? Sorry, I mean an amino acid, which is a monomer of a protein, right? An amino acid will always have an amino group, of course, and a carboxyl group. And then connected to both of these functional groups, we have a central carbon known as the alpha carbon, which has a hydrogen attached to it, and the R group, which could be one of 20 things. 20 different things. That's 20 amino acids that you don't need to know for this class, but if you're taking biochemistry in the future, you will need to know. But don't worry about that right now. So what do these do, right? So why do we care about these two functional groups? Well, they can do something really special. They can become charged. So it's really important that you recognize this. The function of a carboxyl and an amino group is that they can become ions. They can either gain or lose a proton. So when we're losing a proton, right, something becomes negative because we're losing positive charge. And when something gains a proton, it's becoming positive because we're gaining positive charge. So when we're talking about amino acids in our bodies, the physiological solution, the physiological pH is around 7. So that's like pretty neutral, right? So when we're talking about the physiological pH, it's really important that we recognize that an amino group will become protonated, and a carboxyl group will become deprotonated, otherwise known as gaining and losing a proton, respectively. So, why do we care about this? Well, this indicates that this molecule is incredibly polar. This molecule is is not only polar but it's charged. So we can create ionic bonds with so it could be attracted to something like sodium, for example, or on this side, or chlorine on this side, right? So these become charged. And what does this mean? It means that the amino group is basic because it's gaining a proton, and the carboxyl group is acidic because we're losing a proton. Another name for a carboxyl group is a carboxylic acid. So acid, when something's an acid, that's how you know it's losing a proton. So we care about these function groups a lot because they will always serve a specific function within a molecule. So let's go over again, right? We don't care about sulfhydryl. That's like we don't even consider it right now. Methyl group is non-polar, so so when something's non-polar, it's gonna want to be with other non-polar substances, right? So one more thing. I just want to interrupt myself for a second. As a general rule, like dissolves like. I'm gonna put that in like red too. Let's put that in red. Like dissolves like. I want to emphasize this a lot. Why do I want to emphasize it? Because polar substances are always going to mix with polar substances, and non-polar substances are always going to mix with non-polar substances. It's that's just the rule, right? So it's something I really want to emphasize. Non-polar with non-polar, polar with polar. And now when we're talking about charged, with charged that's different because with charges opposites attract, of course. So positive attracts negative, right? But they're still both charged. So charged substances are going to want to be with either polar or charged substances because it has to do with the electrical attraction. So as a rule, like dissolves like. So when something is non-polar, it's going to want to be with something non-polar, right? If a molecule is non-polar, like let's say methane, it's going to want to be with other methane molecules. It's not going to want to be around water, right? Because water is a very polar substance. These two really don't have anything in common. So they're not gonna want to hang around each other. So like dissolves like as a general rule. So 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 yeah, methyl group is a non-polar group. It has carbon, hydrogen, hydrocarbon. That's polar, right? So then moving on to phosphate. How about a phosphate? Phosphate's very charged. It's very much capable of not dissolving, but very much capable of mixing with other polar substances like water, right? Another thing a phosphate can do is create a phosphodiester bond in nucleotides, but we'll get to that later. How about a carbonyl? Carbonyl is able to make hydrogen bonds with water because of this a double bonded oxygen. How about a hydroxyl? A hydroxyl's main purpose is to make something polar, right? Whenever we see a hydroxyl, that's how you know something's going to be polar. And carboxyl and amino, two very very important functional groups. These are both going to either gain or lose a proton, and they're going to become charged. And you can see that up front, first and foremost, in an amino acid. So that was a review of your functional groups, right? Very important to know for this exam. And it's important that you know the functions too, and what purpose they serve. So that was a bit of a long tangent. So let's go back to the question. Hydroxyl groups are the primary chemical group found in a particular biological macromolecule. Which of the following statements regarding this macromolecule is true? So B, it's unlikely to dissolve in water. That's not right. That's wrong, right? It's unlikely to dissolve in water. Well, what did we just learn about hydroxyl groups? Is that they're polar, right? What is water? Water is a polar substance. So like dissolves like, it's going to be able to dissolve in water. So that's not true. How about it's likely to dissolve in non-polar solvents? The same story. If something has a hydroxyl group, then it's going to be polar. I say that with an asterisk, right? There are exceptions to that rule, of course. But if something has a lot of hydroxyl groups, like a sugar, for example, it's not going to want to mix with a nonpolar solvent because if something's a nonpolar solvent, then it's of course going to be nonpolar, right? It's not going to want to mix with something that's polar, so because like dissolves like, right? So these two we can just ignore. How about A? It's likely to form hydrogen bonds with water. Absolutely, right? If some let's look at a hydroxyl. So hydroxyl, right? We have this positive, partial positive charge in the hydrogen and this partial negative charge on the oxygen. So what's this going to do? Well, when we have another water molecule, it's going to make a hydrogen bond, of course, because we have a partial negative charge on the oxygen. So it's likely to make hydrogen bonds with water. That's absolutely correct. So yes, that's number 4. Very long tangent over. Let's move on to number 5. Oh, whoops, sorry. So number 5 is asking, which of the following membrane proteins tend to lack hydrophobic regions on their surfaces? So, A, integral membrane proteins, B, transmembrane proteins, C, integrins, or D, peripheral membrane proteins. So let's go over our proteins then. So what does our lipid bilayer look like? And that's a rhetorical question because I know none of you can talk through your screen right now. What do lipid bilayers look like? Well, they consist of two things. So let's look at an individual component. This is an individual component. This is what's known as a phospholipid. What does a phospholipid consist of? A polar head. Let's see, a polar head. That's a pole, and then a nonpolar tail. Two nonpolar tails. So when we have a bunch of these, what they'll do is something very special. They'll orient themselves like this, right? So a bunch of these, and you create a bilayer. What's known as a bilayer. So what does a bilayer do? A bilayer has a ton of different functions, including but not limited to keeping what's inside the cell inside of the cell, right? So like it's keeping stuff from spilling out. It's able to. It's a semi-permeable. It's a semi-permeable substance, not necessarily substance, but it's semi-permeable, so things can go in and out. It can contain membrane proteins. What else can it do? It do a lot of different things, right? That's just a few functions. So this represents our bilayer. I'm kind of shorthanding it right now because I don't really want to draw a whole lot. Let's draw some of our proteins, right? So this is going to be an integral membrane protein. Why is it integral? Because so this is our protein, right? Why is it integral? Because it's integrated within the bilayer itself. How about this? Well, this is going to be a transmembrane protein because it's going across the membrane, hence trans, meaning across. Well, how about integrin? Well, integrin, that's kind of the same thing as both of these, right? Because the integrin, I mean, judging from the name, integral, they're integrated within the bilayer itself, right? So integrin, integral, you get the idea. How about peripheral membrane proteins? Peripheral membrane proteins kind of look like this, right? They kind of hang out on the polar head of the bilayer. So they're kind of they're not integrated within the membrane itself, but they kind of hang out on top. So I'm gonna draw an orange that's like kind of glued onto there. So it's important to note, right? On the outside we have water, and that's on both sides, right? We have water or some otherwise aqueous substance, right? Whether that be the extracellular matrix or the cytosol, what have you, it's going to be comprised of mostly water. Well, how about the inside? The inside is nonpolar, of course. The inside of this is going to be nonpolar. So what does that mean, right? It means that stuff that something like something that's positively charged, especially like an ion, is not going to be able to go through. But that has more to do with like the that has more to do with like what goes in and what goes out of a membrane, which we're not really talking about right now. But just know that these heads, what's on the outside is polar, and what's on the inside is nonpolar. If something's going to be on the inside, if something's going to be integrated within a protein, sorry, if something's going to be integrated within the bilayer, the middle portion has to be nonpolar, right? So let's use yellow as indicating nonpolar. So the inside of this protein is going to have to be nonpolar. Well, why is that? Right? Well, because we're going back to what we said, like dissolves like. So what does like dissolve like mean? It means that this middle portion, these middle portions of these proteins need to be nonpolar. Well, how about these outside portions? Well, they have to be polar, right? Because they're facing water. And again, like dissolves like, so these have to be polar. And what do we know about amino acids? Well, amino acids tend to be very, very polar. But these inside portions, we're gonna have R groups. So these are gonna be R group interactions. We're gonna have R groups such as phenylalanine, right? Or we're gonna have R groups such as valine or isoleucine. That's not isoleucine. Isoleucine. That's just regular leucine, actually. Isoleucine. Sorry. That sort of thing, which you don't need to know these specifically. Sorry. I love amino acids. I've memorized all 20 of them. So sometimes I like to flex my knowledge in that because it took a while to learn. Anyway, these inside so the R groups on these inside portions are going to be nonpolar, of course, whereas the ones on the outside are going to be polar. So what's the question asking? Which of the following membrane proteins tend to lack hydrophobic regions on their surfaces? Well, of course, these two are gonna have these integral proteins, these integral membrane proteins, these transmembrane proteins, and these integrins are going to have hydrophobic regions on their surfaces, right? Because they have because to be integrated within the membrane, they have to be hydrophobic because the inside of the membrane is hydrophobic or otherwise nonpolar, of which you could use those terms interchangeably. Well, they both mean a little different things. It depends on the situation. Anyway, so what's gonna lack hydrophobic region? Something that's not within the something that's not within the membrane. So what is that? Peripheral membrane proteins. So the answer is D. So what does peripheral mean? Peripheral means on the outside, right? Outside of the main focus. So peripheral membrane, that's going to be the outside of the membrane. Something that's outside the membrane is going to be polar. So what's going to lack hydrophobic regions? Something that's predominantly polar. So that's number 5. Moving on to number 6. Sorry, one second. Got to pull it up real quick. So, number 6 is asking, how do unsaturated fatty acids help keep any membrane more fluid at lower temperatures? So, A, double bonds form kinks in the fatty acid tails that prevent adjacent lipids from packing together tightly. B, the double bonds result in a shorter fatty acid tails, therefore thinner membranes. C, unsaturated fatty acids are more polar than saturated fatty acids. And D, unsaturated fatty acids have a higher cholesterol content than that prevents adjacent lipids from packing together tightly. So this question doesn't necessarily feel like it's meant to mislead you, but it's throwing a lot of vocabulary at you. So this is most easy to know. This question is most easy to answer if you know what a fatty acid is. So let's draw a fatty acid. So in organic chemistry, whenever you have something that looks like this, right? Each point, each apex represents carbon and hydrogen, right? So let's expand this. So this is going to be CH3 with CH2 with CH2 and then etc. Right? So whenever we draw sort of a zigzag shaped like that, that's how you know it's hydrogen and carbon. So what does a fatty acid tail look like? So let's draw a glycerol. So glycerol is what keeps together fatty acids, so to say. Fatty acids could, of course, exist on their own. That's how you get something like soap or soapy substances. But anyway, this is a glycerol, right? This is your glycerol backbone. Glycerol. Okay, so when we have glycerol, what happens is glycerol can attach many different things, but in the case of something like a triglyceride, it can attach 3 fatty acids. Sorry, that's kind of a shoddy work, but anywho. So what is this, right? So this is your glycerol, and these are your fatty acids. So look at your fatty acids, right? What can you tell about them? Well, they're mostly hydrocarbons, right? They're mostly hydrocarbons. So what does that mean? It means that it's nonpolar, right? Because the bond between carbon and hydrogen is a nonpolar covalent bond. So this is not going to mix with water. I mean, of course, you see that with oil and water, for example. Oil does not mix with water. No matter how hard you try, by themselves, oil and water will not mix. Now, if when you get an emulsifier, that's how you know they will mix, but we're not talking about that. Oil and water by themselves will not mix because these fatty acid tails, these are nonpolar, and they're not going to mix with water. And also, look at this molecule, right? Look at this molecule. Like what we discussed earlier, right? It has these carbonyl groups. These carbonyl groups are partially charged. They are partially polar, but that's not what we're talking about. Look at the rest of the molecule, right? This rest of the molecule is non-polar. So when we have these very, very tiny portions of the molecule that are polar, but the rest of the molecule is non-polar, well, the entire molecule is going to be non-polar, right? So anyway, this is what's known as a triglyceride. I'm gonna type it, not write it. This is a triglyceride. So triglyceride, tri referring to three fatty acid tails, glyceride referring to the glycerol backbone. So the glycerol backbone is something that we don't really pay too much attention to because it's not something that necessarily changes from between triglycerides or phospholipids. Anyway, what I've drawn here is that these 3 fatty acids are are saturated. So what does saturated mean? Saturated, saturated. I definitely spelled that wrong. Hold on. Saturated equals no double bonds, and unsaturated equals double bonds. So what does this mean? So when something has no double bonds, if we're talking about fatty acids, when something has no double bonds within the fatty acids, so you don't see any double bonds here, 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 right? Then that means it's saturated, right? No double bonds, saturated. How about when something's unsaturated? So let's delete this a little bit. So let's draw it if it is saturated. So what does that look like? It looks like that. So now it's going out, and now we have this double bond right here. So this double bond here, what this is doing is that's creating a kink. This is going outward like this. So now that it's going outward like this, right? This fatty acid is therefore not gonna be able to pack as tightly together with other fatty acids. So what does that mean? Right? It means that it's not going to be as dense. It's going to be liquid at room temperature. Something like oleic acid, otherwise known as olive oil, is liquid at room temperature, of course, right? So if something's liquid at room temperature, if a fat is liquid at room temperature, or a lipid is liquid at room temperature, it's going to contain these double bonds. It's going to be unsaturated because it has this kink that's going outward like this. It's not going to be able to pack together tightly. So we have another fatty acid. Oh, well, it's like it's not going to be able to tie together like that. So these represent fatty acids, of course. It's not going to be able to pack tightly together. So, anyway, so the question is asking, how do unsaturated fatty acids help keep membranes more fluid at lower temperatures? Right? So going back to this, right? Going back to this is fluid at lower temperatures. Right? Why is it fluid at lower temperatures? Well, because it's not as dense. It's not able to compact tightly together. So of course, if you lower the temperature enough, it is going to become a solid. Yes. However, it's gonna be a much lower temperature than that of a saturated fatty acid, right? So anyway, let's look at the answer. Let's look at the answers again. The double bonds result in shorter fatty acid tails and therefore thinner membranes. Well, shorter fatty acid tails and double bonds don't necessarily correlate, right? So this fatty acid tail, despite having a double bond, it can go on for it can go on for way longer if it can, or it can go on for way shorter. Those two are not mutually exclusive. They don't really have anything to do with each other. So double bonds result in shorter fatty acid tails and therefore thinner membranes. Don't even worry about that. That these don't have anything to do with each other. So how about unsaturated fatty acids are more polar than saturated fatty acids? Well, that's obviously not true because both saturated fatty acids and unsaturated fatty acids are both nonpolar, right? They're like barely polar. We don't even worry about that when we're talking about saturated versus unsaturated. That we're not even referring to a polar region of the molecule, which would be this technically, which would be this region. But we're not talking about that. We're talking about whether or not it's saturated. If it's saturated versus unsaturated, they're going to be the same level of polar versus non-polar. So we don't even want to worry about this. This is just the most incorrect. Well, that's say. Sorry. You got to calm down. So we don't even want to worry about that. How about D? Unsaturated fatty acids have a higher cholesterol content that prevents adjacent lipids from packing together tightly. Well, do you see a cholesterol here? What does cholesterol look like? Cholesterol has like a sort of like multi-ring structure. Well, maybe I could use this. Yeah, there we go. Yeah, this is real organic chemistry right here. Maybe I could rotate it a little bit. Can I rotate it? Will it allow me to? Sorry. Anyway, I'm distracted. Okay, well let's just fill in the rest with it ourselves. So cholesterols and fatty acids are totally separate from each other. This is a cholesterol molecule. It has a bunch of other stuff on the top here. This is a cholesterol. Does this look like a fatty acid? Do these look anything alike? No, right? So unsaturated fatty acids have higher cholesterol contents than that. We're not even talking about this. So normally this question would refer to will refer to the bilayer, right? But we're not even referring to a bilayer. We are so far from bilayers right now. So we are not even worried about cholesterol. So this one is like also extremely incorrect. So C and D we could just like completely throw out. And as for B, that's also incorrect. That leaves A. What does A say? So A is saying the double bonds form kinks in the fatty acid tails that prevent adjacent lipids from packing together. Well, that's exactly right. Yeah, that's exactly what we talked about. The double bonds form kinks in the fatty acid tails that prevent adjacent lipids from packing tightly together. This double bond here, and the fact that it's going out like this instead of going straight like this. Excuse me. This fact is the entire reason why they're unsaturated and saturated fatty acids, whether they're fats that are solid at room temperature and fats that are liquid at room temperature. This is the reason why. So that's your review on fats, so to say. We're moving on now. I sorry, I've been yeah. Anyway. So, question 7. Oh my god, it's been like 40 minutes. We're only talking about question 7. Anyway, so what does question say? What does question 7 say? Which functional group is made up of only carbon and hydrogen? Amino group we can amino group we can eliminate, carbonyl we can eliminate, hydroxyl we can eliminate, and phosphate we could totally eliminate. This one's like the most wrong. Methyl group. What is methyl group? This one you just have to know your functional groups. So we went over the functional groups already, right? So this one's just going to be hydrogen with carbon. That's your methyl group. So that's over. We're done with that one. That one's really simple. So question 8. Chitin and cellulose are examples of. So now we're talking about now we're talking about saccharides, right? So when we're talking about saccharides, there's a few different types. Monosaccharides. One second. Monosaccharides, disaccharides, polysaccharides. Oh, sorry, I'm missing one. Oligosaccharides and polysaccharides. So, what does this mean? Well, if you look at the prefixes mono, di, oligo, poly, well, for one, oligosaccharides we don't even talk about those in this class, right? So don't even worry about oligosaccharides. But what do these mean? Mono means one, di means two, oligo means few, and poly means many. So just from that, right? One, two, few, many, we can determine what's going on here. And so, what does saccharide mean? Saccharide is referring to sugar. Saccharus, I believe, saccharus means sugar in Latin. So a lot of these words come from Latin, of course. That's just never mind. So monosaccharide, one sugar. Disaccharide, two sugars. Oligosaccharides, we don't even talk about that, but few sugars. Polysaccharides, many sugars. So when we're talking about mono and disaccharides, these are monosaccharides is a monomer, right? Monosaccharides are a monomer. Monomer. Sorry. What does that mean? Well, it means that multiple monosaccharides will make up a polysaccharide. So monosaccharides include something like glucose, fructose, galactose, I believe. You don't need to know the difference between those, but they're all sugars and they're all monosaccharides. And if you build up a bunch of them, they'll create some interesting things, right? How about disaccharides? Disaccharides include something like sucrose, which is what you use in your coffee. That's table sugar, of course, and other things. Disaccharides just means two sugars together, which we don't really care about in this class. Oligosaccharides, those are important when it comes to cell signaling. Oligosaccharides are sugars that exist outside of the cell membrane, but we don't really talk about those. And how about polysaccharides? Well, polysaccharides, those are the ones that we care about the most. So what are polysaccharides? Polysaccharides can be a few different things. Either polysaccharides could be structural. They have a few different purposes of what I'm trying to say. They could be structural, or they could be energy storage. So polysaccharides fall into two different categories: structural and for energy storage. And these can translate further to what kind of bonds do they have? Do they have rather what kind of glycosidic linkages do they have? So whether that be alpha 1 4 glycosidic linkage, or beta. Oh, sorry. Whoops. Beta 1 4 glycosidic linkage. So when something has, so as a general rule, something that has alpha 1,4 glycosidic linkage is going to be for energy storage, and something that's beta 1,4 is going to be for structural. So what does this mean? So structural sugars are going to include things like chitin, cellulose, amylopectin, I think. How do you spell that? Amylopectin. So all of these are sorry, all these are structural sugars. All these are structural polysaccharides. So chitin that consists of like something like something like a bug's exoskeleton. Cellulose will consist of like that's like a plant's cell wall, and amylopectin that has to do with like I don't know fungi or something. I don't really quite know that. I don't really quite remember. But energy storage, there are two that we care about a lot, right? And that's starch and that's glycogen. And starch exists in plants. And glycogen exists in animals. But these are energy storage, right? These energy storage molecules typically consist of alpha 1,4. So alpha 1,4. So let's discuss more differences. Alpha 1,4, that's going to be highly branched, easily digested, and beta 1,4 is going to be highly structured slash what's another word? Highly structured or organized, and it's going to be difficult to digest. So these are the reasons why we can't eat a tree, for example. For example, the bark in a tree, very, very, very high in cellulose, very structural, very sturdy. Something like an oak tree, very, very tough. So this is going to consist of something like cellulose. That's in plants. So cellulose is in plants, right? So these are examples of structural polysaccharides, which consist of beta 1,4. So beta 1,4 will look something like this. So this isn't a hydrocarbon, but I just want to shorthand this. And it's going to be they're going to be like kind of linked together by hydrogen bonds. A lot of hydrogen bonds. Like we tend to think of hydrogen bonds as being weak, but when you have a lot of them together, well, they're going to create some pretty strong linkages. Whereas alpha 1,4 are like kind of like branched off like this. So this is going to be starch and glycogen. So we can consider this to be your sort of this can be considered your sort of sugar cheat sheet, so to say. So refer to this if you're ever confused about sugars or polysaccharides more specifically. So going back to question 8, chitin and cellulose are examples of D, structural polysaccharides. Storage. So we're not even concerned about mono or disaccharides because these are not those, right? And we're talking about polysaccharides in this example, and they're not going to be storage polysaccharides because there's only two of those: starch and glycogen, right? So that's question 8. Moving on to question 9. This is another like kind of vocab question, so to say. Um, So it's asking, what is a triglyceride? A lipid that is one glycerol and three fatty acids. B, a carbohydrate made of three sugars. C, a polypeptide made of three amino acids. D, a lipid of one glycerol and two fatty acids. Or E, a lipid of two glycerols, a fatty acid, and a phosphate group. So we just went over a triglyceride, right? So what is triglyceride? I'm gonna pull up a picture of a triglyceride. Actually, I don't want to try it again. Sorry. If you'll give me one moment. So this is a triglyceride, right? So this is your glycerol. This group is your glycerol, and then these are your, and then this, and this are all your fatty acids. And this one is going to be unsaturated because look at here, we have a double bond here, right? This one's going to be unsaturated. This one's going to be saturated. So look at this, right? What does this look like? Well, this doesn't look like a sugar. This doesn't look like a carbohydrate. We can eliminate B. This also doesn't look like a polypeptide. We can eliminate that too. So what is this? Glyceride referring to one glycerol. Tri referring to three fatty acids. So what is that? What does that description fit the most? A lipid made of one glycerol and three fatty acids. So a lipid made of one glycerol and two fatty acids, that needs to be more specific. This is going to be wrong because then if we're talking about just two fatty acids, then there's something else that needs to occupy this last glycerol, this last linkage within the glycerol. And a lipid made of two glycerols and a fatty acid, a phosphate group, that's just intentionally confusing. That one's just like the most wrong, I guess we could say. So what is a triglyceride? One glycerol, three fatty acids. So moving on. I mean that question is really easy if you know what a triglyceride is. So one more thing I really want to emphasize is to know what what poly sorry sorry, I forgot what I was going to say. Something that's really important to know for the exam is to know your polymers. There we go. That's the word I was looking for. It's to know your polymers. So it's good to know the differences between amino acids. Sorry, it's good to know the difference between polypeptides, aka proteins, carbohydrates, aka polysaccharides, nucleic acids, and lipids. It's important to recognize all four of these, in which there's a question on the practice exam later about that. So, what is question 10 asking? Every protein has a unique shape and function because A, the number of nitrogen atoms in each amino acid differs. B, the bonds between the amino acids varies from protein to protein. C, each protein has a unique sequence of nucleotides. Or D, each protein has a unique sequence of amino acids. So, I'm just going to say the answer. The answer is D. So proteins are really, really awesome. What does your amino acid look like? I'm going to draw the charged version. In fact, I'm going to draw it like this. Okay, this is your general amino acid. So simple, so humble, yet so beautiful. Over here, we have our two functional groups, the amino group and the carboxyl group. And over here, we have the central alpha carbon. And over here, we have the R group. The R group is what changes, of course. It can be one of 20 different things. So this is your amino acid. So the number of nitrogen atoms in each amino acid differs. Well, let's look at this. Excuse me. Sorry. Anyway, so this portion of the molecule, this R group portion, this is what's going to change. But this portion is known as this portion is known as the amino acid backbone. This never changes. If this changes, then it's no longer an amino acid. All right? So this never changes. I want to emphasize that. And the same thing with a nucleotide, which we'll go over later. So if this never changes, right, the number of nitrogen atoms in each amino acid will never differ. It's always going to be just one. If it said the number of hydrogen atoms, that would differ, because this amino acid is lacking a hydrogen over here, and it has an extra hydrogen atom over here, right? But the number of nitrogen never ever changes. Never, never, never. The R group is what changes. And the R group may contain some nitrogens or may not contain nitrogen, but the backbone never changes. So, besides, even if it did change in the R group, that wouldn't necessarily encompass what's different about each amino acid, what's different about each protein. So how about the bonds between amino acids varies from protein to protein? That's also wrong, because let's expand our protein here. So when we link together multiple amino acids, right? That's when we get a protein, because amino acids, of course, are the monomer of a protein. So let's do this. Then yellow for the R group. So what I've drawn here is a polypeptide, right? And let's recognize this. So this right here, this is a magical thing. In fact, I'm gonna highlight it in bright green. How about that? So this right here is a magical thing. It's what's known as a peptide bond. So this is your peptide bond. Peptide bonds are what link together amino acids. So if you look here, this is one amino acid, and then this is another amino acid. And what do? They share this nitrogen, right? They share it via this peptide bond. So this peptide bond is what links them together. We add amino acids via peptide bonds. We link them together one by one by one by one by one. And when we form a peptide bond, this is known as a dehydration synthesis, the dehydration reaction, and we're losing water as a result. Right, because we're dehydrating the molecule. So, this is a peptide bond. So this peptide bond does not vary either, right? This peptide bond will always be the same. This peptide bond, if we link together more, we're gonna create more peptide bonds, and the peptide bond will never change. It will always be a peptide bond because it's linking together a peptide. It's linking together amino acids. So B is going to be wrong also, because the bonds between amino acids doesn't vary from protein to protein. How about each protein has a unique sequence of nucleotides? Well, that's wrong, because nucleotides have to do with nucleic acids. We are very far away from amino acids. Sorry, we're very far away from nucleic acids when we're talking about proteins. So we don't even want to worry about C. C is that's the most wrong. So D, each protein has a unique sequence of amino acids. Well, let's look at this. Like I said, these R groups can vary. So let's draw out some R groups. Yes, let's do that. So if we delete these R groups could be one of 20 things, as I've discussed. So How about this R group can be, let's make this one isoleucine. And actually, that's wrong. Isoleucine. And let's make this one phenylalanine. Phenylalanine, right? Both of these are polar. And both of these are very different from each other. This one has a ring structure. This one does not have a ring structure. Very different from each other, right? But how about instead of phenylalanine, I drew just an H. This is known as glycine. This is known as glycine, and that this can exist too. This is also an amino acid. But how about CH3? This is known as alanine, also a nonpolar substance. How about just an OH? This is known as serine. These can change. These will change, and that's what makes them so unique. They could be one of four different categories. These side groups can either be nonpolar, polar, positive, or negative. So what does a positive and negative one look like? Well, let's draw glutamic acid, otherwise known as glutamate, right? So, and then it has this carboxyl group at the end, and it's going to be negatively charged. This is what makes your food so delicious. This is glutamate. Otherwise, and when you add a sodium to this, it becomes monosodium glutamate, otherwise known as MSG. That's what makes food so delicious, right? MSG and salt. So that's what a charged amino acid will look like when it has a charge like this, or when it has a like sort of amino group and then it can gain a proton. So negative, positive, polar, nonpolar. Those are your four types of amino acids. So the answer is D, right? So this is going on for a lot longer than I anticipated. Um, so yes, I'm sorry about taking so long, and there's a lot of exam left. So I could hurry this up a little bit, in which I will do so now. Well, except for this next question. This next question is very important, I think. So the question is asking alpha helices and beta pleated sheets are examples of primary, secondary, tertiary, or quaternary structure. So right off the bat, I'm going to answer it. It's secondary structure, right? But that doesn't help you, so we're going to go over it. So primary structure of an amino acid, that's going to be your amino acid sequence. Right? And I'm just going to write them all out as they come along. Then we have secondary structure, tertiary structure, and quaternary structure. So secondary structure is going to be backbone interactions. Excuse me. Tertiary is going to be R group interactions. And quaternary is going to be protein interactions. Or even more specifically, interprotein interactions. Inter meaning between. So, what do these mean? So, amino acid sequence, right? The amino acid of something, so each amino acid has a one letter code. So, what this can mean is you can have an amino acid with A, G, H, not H, I, P, L, I, P, A, P, T, Y, or it could be like Y, P, L, I, P, A, S, T, or it could be um, V, A, or it could be that. So what I'm trying to get at is all of these are different from each other. But each of these proteins will be one specific protein, right? They're all different from each other, and that's because of the amino acid sequence, right? Remember, the R group is what changes. So because the R group is what changes, each of these are going to be different from each other. They're going to fold differently. So the very basis of a protein folding is the amino acid sequence. So how about backbone interactions, right? Backbone interactions include two different things: alpha helices and beta pleated sheets. And so what do these mean? So what? So when we're talking about backbone interactions, we're referring to the amino acid backbone interactions. So if going back to the amino acid backbone, so going back to the amino acid backbone, right? You have an H here and a double bonded O here. So what do these have in common? Well, these can make hydrogen bonds. Well, this bond is non-polar, right? But this is still a hydrogen, is it not? Right. So an oxygen, a double bond oxygen in a different amino acid can still come in and make a hydrogen bond, just like the the hydrogen in a different amino acid can come along and make a hydrogen bond with this oxygen, right? So when we have this on a larger scale, it creates one of two different things. It creates either an alpha helix, which looks kind of like this, an alpha helix, or it can create a beta pleated sheet, which looks like this, kind of. I could pull up a different image so you can have a better idea of it. Hmm. If you'll excuse me for one moment. Okay, yes, this is a good example. So I just realized I've been saying it wrong the entire time. So this is not the hydrogen. This is not the hydrogen that we're worried about. This is wrong, right? This is not going to have interactions. What is going to have interactions is the H on N, though. But anyway, right? So beta pleated sheets kind of look like this, right? They kind of look like this. They kind of fold together like that, and then alpha helices come along and they create like a sort of loop. So that's alpha, beta. That's alpha and beta, right? So then we go into R group interactions. So R group interactions, right? Going back to like what we talked about, there are four different types of R groups: non-polar, polar, positively charged, and negatively charged. So what do these mean? It means that polar R groups are going to interact with polar R groups. Non-polar R groups are going to interact with non-polar R groups, like dissolves like, as we said. And then positive is going to be attracted to negative, and vice versa. So when we take all of these into consideration, we can create our protein, right? So let's pull up an example of a protein. Let's try and use a like a simple. There we go. So this is our protein then. So look at this, right? What do we see here? So let's use orange. What do we see here? We see there's an alpha helix here. There's a beta sheet. No, wait, no, sorry. There's a beta sheet hiding within the green here. There's another beta sheet here, and these are all interacting with each other, like like here, for example, they're interacting. Sorry, here they're all interacting. Here they're all interacting. They're kind of folding in on themselves, and this protein will always, within a certain temperature and pH, it will always fold like this. And what determines this? The R groups. The R group interactions determines this. And a protein will function in this conformation, right? So we have different groups. So red would represent like a polar sequence of amino acids, or blue would represent a non-polar, or yellow, that sort of thing, right? So this is your R group interactions. That's how proteins like fold on an even grander scale within the R groups. So that's tertiary structure. Another thing we must note is that from tertiary to primary, all proteins do this. All proteins. This is all proteins. Whereas quaternary structure, this is only some proteins. So let's pull up another example, right? Let's pull up a hemoglobin. So hemoglobin exists in your blood, of course. So hemoglobin is how oxygen is able to be carried from cell to cell via your blood cells, right? So look at this. Excuse me. So each one of these, so so each colored region is a different protein, and we call these subunits. We call these subunits, right? So each one of these are different proteins. So this orange one is one protein, this brown one is one protein, this purple one is one, and this pink one is one. But the difference between this protein and the one I showed you previously is that the previous one can function on its own, right? The previous one can function entirely on its own, whereas this one, right, this one, it functions as a whole. Each one of these subunits cannot function by themselves. It needs to function as a whole. So when they function as a whole, that's how you know it's interprotein interact. I just realized I spelled interactions wrong. Interactions. There we go. Interprotein interactions, right? That's quaternary. So yes, these are all. So when something functions as a protein as different subunits, that's quaternary folding. So yes, that's number 11, and I'm getting kind of tired and kind of fatigued. So I'm gonna like do a sort of lightning round, so to say, for the next few. I'm at least gonna try. If I think something's important, then I'm gonna note it, of course. So two chains of nucleotides in a DNA molecule are held together by hydrogen bonds. So let's look at a picture of a DNA molecule. Right. This is just something that you kind of have to know. So DNA molecule. Let's use this one actually. This one's good. Okay, so this is your DNA molecule. So let's look at what's going on here. So these blue portions represent the phosphates. So the phosphates are what link together nucleotides. So a nucleotide has three primary components. Actually, let's go ahead and just shorthand it here. There we go. Isn't MS Paint such a beautiful thing? All right, and let's put the phosphate in yellow. So nucleotide, or I'm going to draw DNA because DNA is the one that we care the most about because that's of course. Okay, anyway. So this is DNA. How we know it's DNA? Because it's deoxyribose. We're missing an oxygen here on the 2' carbon. So when we're talking about another thing, when we're talking about carbons, we number the carbon. So each one of these apexes, like I said, are a carbon with hydrogens. So we number them 1, 2, 3, 4, and up here there's 5. We number them going clockwise, right? So let's see what's going on here. So in this portion, so on the 2', so anyway, in DNA, 2' carbon has a hydrogen instead of an OH. This is your sugar group, this is your phosphate group, and this is your nitrogenous base. And I'm I know this is kind of confusing. There we go. This is your nitrogenous base. So nitrogen is referring to the fact that it's nitrogen containing. So each one of these contains some portion of nitrogen, and then they'll have a methyl group over here, then other stuff, and then anyway. So for example, this would be guanine. So of course we have A, T, C, and G, and remember your base pair rules. This is very important to note. I just want to say, very important. A always pairs with T, and C always pairs with G. Otherwise, adenine always pairs with thymine, and cytosine always pairs with guanine. So what's going on here? Why are these paired together? Well, look here, right? Well, we have 2 DNA molecules, one that's running this way, and then another one that's running this way. This is known as anti-parallel. When they're running opposite directions, what can happen between these 2 bases is that they could actually pair together using hydrogen bonding. So if you notice here, it's a sort of dashed line. Dashed lines almost always represent, in chemistry anyway, hydrogen bonds. So despite the fact that you can't see them here, there are hydrogens and nitrogens and oxygens that are interacting with each other, creating H bonds between amino acids. So when 2 chains of nucleotides and DNA molecules are anti-parallel, they're being held together by hydrogen bonds. Not covalent bonds, because that would be way too strong. Not hydrophobic interactions, that would be way too weak. Not ionic interactions, because there are no ions. And not peptide bonds, because we're talking about DNA, not proteins. So that's number 12. So, okay. I'm just gonna wait one sec. Okay, so number 13 is asking, what type of bond is indicated by this arrow? We already went over this. This is a peptide bond. Remember the bright green bond that we discussed when we have carbon connected to the nitrogen. Yes, this is a peptide bond, and you could determine this because phosphodiester. Well, there's no phosphorus, right? Phosphodiester occurs between nucleotides, and it's not glycosidic linkage because glyco means sugar, and you don't see any sugars here. So, process of elimination, it's going to be peptide bond. So number 14. Number 14 is a bit of a critical thinking question, but we can go over the difference between hydrolysis and dehydration. So, when a liver cell breaks down glycogen into individual glucose monomers, the cell is performing blank reaction. So, when we're breaking down glycogen, so glycogen is what? Glycogen is a sugar, right, involving individual glucose monomers, right? So, when a liver cell breaks down glycogen, that's going to be hydrolysis. So let's have a bit of a lesson in Latin and or Greek. Hydro means water. I'm sure many of you already know that. Lysis means to cut. So putting it together, hydrolysis, cutting with water. That's how you know, right? So what does this mean? So let's draw out two sugars. So imagine, okay, well, hold on. There's an O here, and then there's an OH over here. One moment, sorry. Anyway, sorry about that interruption. Um, anyway, so these are two sugars, right? So these two sugars are held together by glycosidic linkage, right? So when we break this apart and we use water, let's use water in blue. When we're adding water and we're breaking this apart, what is happening here? We're splitting them apart. So this would be known as hydrolysis, right? So hydrolysis, and we have an OH here now, and then an OH here. Okay, but then when two sugar molecules come together, it doesn't have to be sugar either. It could be two amino acids or two nucleotides. When two of them come together, it creates it. The inverse happens. We're actually we're they're forming a bond, and then water is leaving. Hence dehydration. So when we're splitting it, when we're splitting it, that's hydrolysis. When we're forming it, that's dehydration because we're losing water. So moving on. Number 15. Okay. So I think number 15 is a very important one. So let's go over it. So number 15 is asking us number 15 is asking us to recognize um each polymer, not polymer, each monomer. Each sorry, it's asking us to recognize each molecule, right? So these are all monomers, right? So let's look at these individually. So for this one, so this is 15 through 18 actually, sorry. So let's look at this. What is this? Well, we have an amino group here, and then we have a carboxyl group here. What is that, right? That's of course going to be an amino acid, right? So A, right? So it's important that you recognize whenever you see an amino group, a central carbon, and a carboxyl, that's an amino acid, which will make proteins, right? How about this? This is a type. So look at this closely. What does this consist of? Mostly hydrocarbons, right? Look at these. There's only one polar. There's only one polar portion within the entire molecule. So what does this tell you? If something's composed of mostly hydrocarbons, it's going to be mostly nonpolar. The only mostly nonpolar macromolecule that we discussed is, of course, going to be a lipid. Yes, so it's going to be a lipid. So you have to determine now, well, which type of lipid is going to be? Is it going to be cholesterol, or is it going to be a fatty acid? So cholesterol and other steroids are the only molecules that have a multi-ring structure like this one. So a fatty acid, what is a fatty acid? Fatty acids have like the ketone. Sorry, they have the carboxyl group with a tail, right? So fatty acids always have this tail, right? So, but do you see that here? There's not really much of a tail going on. There's just there's just the four ring structure that's mostly nonpolar, so it tells us a lipid and tells us it's not a fatty acid, so it's going to be C, cholesterol. How about a monosaccharide, how about this, right? So we could determine it's one of these. It's either B or E. So it's going to be a monosaccharide. Or it's going to be a polysaccharide. Well, let's look at this. It consists of one sugar, right? So how do we know it's a sugar? If it's going to be in this sort of this sort of hexagonal conformation, and it has a bunch of OHs. So it has OH here, OH here, OH here, OH here, OH here. It has a bunch of OHs, right? And then if we remember, this is our general formula for sugar. So N is the number of carbons. So if we have 5 carbons, it'll be C5H10O5. Or if that's 6, like this one, it'll be C6H12O6. So so this is a sugar, right? We can determine that. How many sugars is it? It's one sugar. So one sugar, what's that going to be? Get further. What's that going to be? A monosaccharide. So how about question 18? Is this going to be what is this going to be? So it has 3 central components, right? So what are components? A base containing nitrogen. We have one nitrogen here, another one here, another one here. So we have a lot of nitrogen. So this is going to be a nitrogenous base. And then we have, what is this? A 5-carbon sugar, right? We have an OH here and then an O in this sort of like pentagonal structure. And then we have a phosphate group. Well, what's the only thing with a phosphate besides a phospholipid? Well, whenever we have these 3 things, that's how you know it's a nucleotide. So it's going to be D, nucleotide. So, as a general rule, I think it's really important that all of you recognize the types of macromolecules. So, the rest is sort of like a written response section. Let's see what I'm gonna do. So I'm not gonna give my own answer. We're just gonna discuss the given answer and just kind of go over like that, right? It's just one of those things you just have to know, unfortunately. So when it comes to written responses on the exam, you're not gonna have to do all of them. You're just gonna have to do like 1 or 2 of them, most likely 2. So let's look at this. Explain why we describe carboxylic acids. Why we describe carboxyl groups as acidic and amino groups as basic. What charges do these acquire? Right. So carboxyl groups, as we discussed earlier, release H plus into aqueous solution in the physiological pH and become negatively charged. So like I said, we discussed that earlier. Right. What is a carboxyl again? This is your carboxyl. So we're losing this hydrogen and it's becoming negative. Right? So every time we lose a proton, something's becoming negative and it's therefore acidic. So it'll become negatively charged. How about an amino group? An amino group removes a proton from solution, becoming this. Right? So that's your so this is your normal amino group. Amino group then becomes this in physiological pH, which we also talked about. So both carboxyl and amino groups exist within within amino acids. So how about describe 2 structural differences between RNA and DNA? So again, let's create like a sort of like. So how about this? Let's do this. Whoops. Let's do this. Yeah. Okay. RNA on one side, DNA on another. So what are these differences? So so RNA, let's just write them down. RNA contains it just contains ribose. Or let's expand RNA. RNA stands for ribonucleic acid. Right? So that tells us that RNA consists of ribose. RNA is single-stranded. And RNA has bases A, U, C, and G, in which A pairs with U and C pairs with G. Right? So let's look at DNA then. What is DNA? Well, DNA is, of course, deoxyribonucleic acid, which consists of deoxyribose, is double-stranded, creates helix, and the bases are A, T, C, and G, in which the base pairing rules are A pairs with T and C pairs with G. So what is the difference between these two? All right. So let's look at the individual monomers. So let's draw out DNA then. So DNA, let's get a little pentagon here. So that's our 5-carbon sugar. And let's draw a red oxygen here. Or actually, I will do one better. How about this? Okay. And then we have the phosphate. And then it's bonded to a nitrogenous base. And then it has an OH on the 3' carbon and an H here. So let's select this. Let's copy it and let's paste it over here. So what is RNA? The only difference between DNA and RNA is this. Watch this. This is your only difference. These look pretty much the exact same, don't they? I mean, you still have the phosphate, you still have the 5-carbon sugar, and you still have the nitrogenous base. Same thing over here. You have these 3 components. But the sugar is what changes. Over here, we have what's known as deoxyribose, as in removing of oxygen in the ribose sugar. So this sugar is known as ribose. Or specifically, this sugar is known as ribose. This one is also ribose, but the only difference between this and regular ribose is that it's missing an oxygen here. So because it doesn't have that, it's known as deoxyribose. So because it's deoxyribose, it's known as deoxyribonucleic acid. So that's the only difference. That's the only difference. And because of this difference in the sugar, RNA is single-stranded, DNA is double-stranded, and then these two, then the base pairs also change. These are the differences between DNA and RNA. So screenshot this if you'd like. Otherwise, I'm going to move on. So we already went over saturated and unsaturated fatty acids. Just remember that one that saturated is more solid, unsaturated is more fluid. For the exam, I don't think you need to know the difference between alpha and beta, which we already kind of went over. Um, we also went over condensation and hydrolysis reactions. We talked about a little bit about cellulose and the different sugars. So actually, I would say we covered just about everything. So yeah, I'm sorry this took so long. Um, I really hope that you do well in the exam tomorrow. We went over pretty much everything that we need to go over. Just one thing I want to emphasize: carbon. So good old humble carbon. Carbon can make 4 bonds. 4 bonds to what? To really anything. Or not anything, but you know, it can make bonds to nitrogen, oxygen. If so, this is vital to know for the exam. What else? So one last thing I want to go over, and I get a lot of questions about this. So here it is: carbon and hydrogen, and then anything bonded to itself. So carbon and carbon, oxygen and oxygen, nitrogen and nitrogen, whatever. All of these are non-polar covalent bonds. All of these are non-polar covalent bonds. So carbon, hydrogen, non-polar. Anything to itself, non-polar. Right? So how about oxygen and hydrogen? Or nitrogen and hydrogen? Or carbon and oxygen? It all has to do with the difference in electronegativity, right? And all of these are polar covalent bonds. Unfortunately, this is just something that you have to remember. But you could remember this easily because, well, hydrocarbons, that's hydrogen and carbon, of course. That's going to be nonpolar because those are primarily, that's what fats are made out of. That's what a bunch of other nonpolar substances are made out of. Right. How about OH? Well, add one more H, and that's water. And water is like one of the most polar substances ever, right? So that's going to be polar covalent, right? So this is just another thing I want to emphasize. It has to do with the difference in electronegativity. How about ionic bonds? When two things are fully charged, like sodium and chlorine, what these will do is they'll create a bond with each other. Not a covalent bond, mind you. It's just that sodium is going to donate its one electron to chlorine, and these are going to make salt. One compound. This is one compound. So other than that, I think I've covered pretty much just about everything. So I'll leave you with this. Thank you for watching. Right. So I hope all of you have a good exam tomorrow. Well, I guess I kind of messed that up. Anyway, I hope all of you have a good exam tomorrow, and I have faith in you and faith that you'll do well. If you watch the entirety of this video, then I've covered just about everything that you need to know for the exam. So please, 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 let me know if I did good well in this, if I did well in this review, if you'd like me to do any, if you would like me to cover anything else in the future, if you'd like me to, just let me know how I'm doing, that sort of thing, right? I just hope all of you have a good exam. So take care, and I have faith in all of you. So goodbye.